the name of the next speaker, Andrés Aldana. Thank you, Andrés, for accepting to do a presentation in the RePo webinar series. I will do a brief introduction to Andrés. So, Dr. Andrés Aldana is a computational biologist and network scientist, currently serving as a postdoctoral research associate at the Barabási lab in Northeastern University, collaborating with the Harvard Medical School. His research integrates network science and artificial intelligence to identify therapeutic targets and repurpose drugs for cardiovascular and other complex diseases. He earned his PhD in biomedical science from the National Autonomous University of Mexico, where he collaborated with world leaders in male reproduction to propose novel targets for contraceptive development and fertility treatments. Dr. Aldana has a strong background in systems biology, computational modeling, and artificial intelligence, and has taught over 20 university level courses in artificial intelligence, pattern recognition, and complex systems. So, Dr. Andrés Aldana will speak about NetMedPy, a Python package for network medicine and drug repurposing. Andrés, the screen is yours, and thank you very much again for accepting to give this talk in the RePo webinars.
Thank you very much, Hugo. You can hear me?
Yes.
Yes, okay, perfect. And you can see my screen, right?
Yes.
Perfect, okay. Thank you, Hugo. Thank you for the introduction, and also thank you for the invitation. It is great to participate with this community. Also, I want to thank to the audience for assisting to this talk. Yes, I want to talk to you about this Python package that we have developed at the Barabási lab over the last year, I think, which is called NetMedPy, and whose purpose is to facilitate the network medicine framework to the expert and non-expert users with a particular focus on drug repurposing. So let me start by telling a little bit what is network medicine and how we use it for drug repurposing. So, get my pointer. There you go. I am quite sure that most of you are familiar with the Osler paradigm. Sir William Osler, which is considered by many as the modern or one of the fathers of modern medicine, stated that each illness is a distinct and definable pathological entity that affects primarily a specific organ or system, and that has a single and identifiable cause, either a pathological agent or a mechanism. It is what we know as the one disease, one organ, and one cause model. And this way to visualize and conceptualize medicine is foundational for modern medicine. It's so important, and it was so successful that it defines not only how medicine is executed nowadays, but also how medicine is taught at universities, and actually how hospitals, for example, are organized. However, there are some diseases, some complex diseases like diabetes, atherosclerosis, depression, cancer, among other complex diseases that doesn't seem to follow that structure. And the problem with those diseases is that we don't have only one disease, but frequently we have interconnected diseases like hypertension, obesity, and diabetes that can co-occur in the same, and that frequently co-occur in the same patient. Most of these diseases affect more than one organ. They have shared molecular pathways. They affect multiple organs. So we don't have this specific organ affected by the disease. And also we have multiple factors for these diseases, like lifestyle, mutations, environmental factors, genetic load, geography, ethnicity, etc. So we have multiple factors that contribute in the development of these diseases. And so we started, given that situation, we started to look at genomics in order to understand and to treat these complex diseases. And in 2003, the Human Genome Project was completed that sequenced the entire human project, identifying around 20,000 protein coding genes, and also identifying genes which mutations are associated to diseases, what we know as disease-associated genes. And the promise of the Human Genome Project was that, or still is, that gene discovery is a path to understand disease. Since the hypothesis is that mutations are the cause of the disease, then identifying the genes mutated for the diseases can lead to cures, and that we can treat diseases in a personalized way rooted in the genome of each individual patient. So the thing is that with the Human Genome Project, we learned the letters of life, but we didn't learn its grammar, because in our view, giving you the list of the protein coding genes and the disease-associated genes is the equivalent of giving you the list of the street names in New York. It is not enough. It is good, but it's not enough. It is not enough for me to tell you how do I get from point A to B. For that, we need a map. We need a map that tells us how the different streets are interconnected between them. But even the map is not enough. We also need the tools to explore that map to convert the data into useful information that we can use in decision making. In this case, how to get from point A to B in the shortest time possible, avoiding the traffic that is very characteristic in New York City. So network medicine aims to fill that gap, except that in our case, the map is very messy, a lot more than in New York City. And instead of street names, what we have are proteins, and the genes that are transcribed and that transform into those proteins. And the interaction between proteins gives place to these very complex protein interaction networks that we know as the interactome. And then the mutations in the genes, the mutations in the disease-associated genes, well, not all of them, but some of them, will modify the protein structure and function. So essentially, we are altering how, or the mutations alter how the proteins associated interact with each other. The mutations disrupt those interactions. And these perturbations propagate through neighboring proteins, and they can spread across the whole system or across other systems that are not related with that particular disease. So the main purpose of network medicine is to integrate this protein interaction and disease gene associations data into a network-based framework that allows us to better understand, diagnose, and treat diseases. One of the main results of network medicine is that the disease-associated genes are not randomly scattered across the network, but rather they form clusters of very tightly interconnected subgraphs that are localized in some specific regions in the interactome, what we know as the disease modules. And in this case, what I am showing here is the asthma disease module. And you can see that for the asthma disease module, there is this cluster here. It's not scattered across the whole network. It is clustered here, and the subnetwork of that module is very tightly interconnected. So not only that, but also one of the important discoveries of network medicine is that the disease modules are also interconnected in the interactome. So we don't have different mutually exclusive disease modules for the different diseases. What we commonly have are diseases with shared mechanisms, and those diseases with shared mechanisms have overlapping or adjacent modules within the interactome. And here I show you an example of the disease module for multiple sclerosis and the disease module for rheumatoid arthritis. And even when they are different diseases, you can see that they share some pathways, they share some molecular mechanisms, and they are close to each other in the interactome. These shared and proximal molecular bases can explain the co-occurrence of different diseases in the same patient and can also explain the pleiotropy of the disease-associated genes. But also having common pathways suggests a potential for shared drug targets, meaning that the drugs that are used to treat one disease are potentially or can potentially be used to treat the other, which is, as all of you know, the heart, the essence of drug repurposing. So these shared mechanisms, the interconnection among diseases is not infrequent. In fact, what we have is that most of the diseases have shared mechanisms with other diseases. And in this graph, each node represents a disease. So we have a map of all the different diseases. And two diseases are connected if they share at least one or more disease-associated genes. So you can see that there is a very tight interconnection among different diseases, and the colors represent classes of diseases. And one thing that immediately pops in the eye is that similar diseases or diseases that belong to the same class tend to cluster together, meaning that diseases that are similar in nature or that affect a similar system have shared mechanisms, which is not surprising. So we have here the group of neurological diseases. We have the cancer, the different forms of cancer. We have cardiovascular diseases. We have dermatological diseases, each one forming its own cluster, which is expected. But also you can see that there are other diseases that not only interconnect with other similar diseases, but they are connected and they share mechanisms with diseases that are not even in the same class. And the perfect example is diabetes. So you can see here that diabetes shares mechanisms with a lot of different other diseases that belong to many different classes. So we can use that information for drug repurposing. And another of the main results of network medicine is that the network distance can predict drug efficacy to treat the disease. The basic principle is that the drugs that are used to treat the disease, the targets of the drugs that are used to treat the disease and that are known to be effective for a disease are also in close proximity and or overlap the disease module in the interactome. And this gives us a framework to predict how effective can a drug be to treat a certain disease. So essentially, what we have, or the main idea is that if we can identify the targets of a certain drug and we can identify the disease-associated genes in the interactome, the main question here is how many steps, if we create a disruption here by using a drug, how many steps does it take that perturbation to reach one of the disease genes, to reach the disease module? And the hypothesis here is that the less steps that we take, the more likely it is that the drug is effective in regulating that disease module or in modifying the activity of the disease-associated proteins. So the framework for drug repurposing is very simple. Let's say that we have many different drugs, each one of them targeting a different group of targets, and we have our disease module located here in the interactome. So we can measure the distance or the network-based distance, which is essentially what is the shortest path between the drug targets and the disease modules for each one of the diseases. We integrate each one of the shortest paths from all of the targets to the disease module, and we evaluate the statistical significance of that distance testing against the random expectation. And then by evaluating the statistical significance, then we can create a priority list where at the top of the list will be the drugs that are most likely to affect the disease module. So essentially what we have is a priority list with the best, or predicting the best candidates of drugs to treat a disease. It is very simple. This is a typical pipeline for network medicine-based drug repurposing. Essentially, we start with many different patients, and then we have to collect from those patients and from the literature, genomic, proteomic, metabolomic, and other omic information. We integrate that information into the protein-protein interaction network, capturing also the disease genes and keeping track of the drug targets. And we validate then the disease modules. Validating here means that we verify that the genes associated to a disease, that the disease module is meaningful, is biologically meaningful. We calculate the distance from the different drugs to the diseases that we are evaluating, and then after prioritization of the different drugs, then we can experimentally and clinically validate the results. Now, from the computational point of view, there are challenges for this framework. The main challenge is that there is a high computational cost, because calculating these distances is very computationally expensive, and then evaluating against random expectation makes it even more complex. So we have limited scalability. Typically, the packages that exist, they don't integrate all the frameworks. So we have to collect pieces of software from here and there. So the programs and the framework is fragmented, and this creates problems in standardization of the mechanisms and the methods. There is a lack of flexibility for custom pipelines, meaning that these packages have pre-built functions and there is little flexibility to modify them, and all of this translates in difficulty to reproduce and validate the results. So all of these challenges led us to the development of NetMedPy. So finally, we reach the part of the software. NetMedPy happens bridging the part of the data, namely the PPI network, the drug targets, and the disease-associated genes, and the validation of it and evaluation phases. So essentially, NetMedPy provides the interface between the data collection, the analysis, and the evaluation part. It is focused on drug repurposing and screening studies, so it is optimized for performance. It works in very large data sets. It almost automatically extracts and validates the disease modules. Is very statistically flexible, meaning that it provides the user the framework for many different statistical tests, corroborating results on their different hypotheses. It runs in parallel for the framework, for the infrastructure that allows it. And it is very customizable. So the user can actually include their own functions to define distances and to define the statistical procedures. So this is a case study that exemplifies the execution of NetMedPy, of this framework. We use the effects, or we evaluate the effects of vitamin D on different diseases. We choose vitamin D because it's very well understood, or better say, is one of the best understood vitamins and drugs that we have. It is known that it has a very good effect on inflammation processes and also in cardiovascular diseases. So we wanted to see if we could recover those effects. So essentially, there are three functions that are fundamental and that are very simple in this framework. This one here is for disease module extraction and validation. So you only get as input the protein-protein interaction network and the disease genes, and it will automatically validate the statistical significance and extract the disease module. Then this step here is necessary for NetMedPy because the way in which NetMedPy optimizes the framework, the whole pipeline, is by keeping track of the distance between nodes in the network, so that this distance is calculated only once and it can be reused later every time that we need it. So the more interconnected we have diseases and the more diseases that we want to test, the scalability is better because then we can reuse the same node pairs or the distance between node pairs over and over again instead of calculating it every time. And this here is where the magic happens. This is the function to screen for the proximity between the vitamin D and the different diseases, which is essentially identifying what are the diseases that are most likely affected by vitamin D. And as you can see, it is very simple. It receives the vitamin D targets, the list of diseases and their associated genes in a form of a dictionary, and the protein-protein interaction network, and the rest are control parameters. And that's it. Essentially, that provides the necessary data to produce this plot. And as you can see here, or I can show you, then the statistical proximity measured by the Z-score of the distance between vitamin D and the different diseases captures actually what I am saying, that inflammation, coronary artery disease, and vitamin D deficiency and asthma that has a very strong inflammation component are among the top diseases affected by this vitamin. So the framework works. This is only to show that this framework works and that NetMedPy can, in very few steps, be used to these kind of studies. Sorry. So beyond shortest paths, NetMedPy offers multiple notions of biological distances, because we know that in biology, rarely the information, the signal transduction and the biological pathways happen linearly and follow the shortest path between proteins. Typically, or in a lot of cases, signal transduction follows probabilistic or weighted patterns. So NetMedPy offers flexible distance measures, including shortest paths, distance based on random walks, biased random walks, and communicability. And it also offers the possibility to the user to define their own notion of distance, allowing the users to specify custom measures. This helps in providing different kinds of information, but also in validating the results, testing under different hypotheses and testing under different ways in which the information can flow across the network. As you can see here, the patterns, even when the distances are different, the patterns are mostly the same. Also, we included in the package different null models for robust network inference. This is because, so the observation here is that the distance between the drugs and the diseases in the interactome is fixed, which is represented here by this black line, which is the distance between vitamin D and I think it was asthma. And the thing is that the statistical test, the statistical significance of this observable distance is different depending on the null model that we present. That is depending on the hypothesis that we are presenting or that we are testing. So for robustness of the results, we included different null models that allow the user to test under many different hypotheses, including also custom ways to provide for null models and different kinds of ways to sample and to test against random expectation. Random doesn't always mean to scatter or to pick nodes scattered across the entire interactome, but rather getting random nodes that preserve certain structure. So NetMedPy allows for that. And one of the main features is the speed. Performance is one of the key features of NetMedPy. So it is very fast compared with other implementations. NetMedPy was designed for high throughput execution. So the largest the data sets, the speed up is better. It was built with parallelization and memory efficiency in mind. And it is efficient even when the module size and the network complexity grows. So in this plot here, I show you the execution time in seconds as the size of the disease modules increases. And yes, in NetMedPy, you can see that the execution time increases, but is orders of magnitude lower than for other implementations that can be found in the literature and online. So NetMedPy is fast, in few words. This work has been published. This is an image of the bioarchive, but the work has been accepted in bioinformatics. It is currently in the process just before publication, but it is already accepted. So you will soon can look for it in the bioinformatics webpage, but you can also download the full manuscript from bioarchive. And as a summary and takeaways, NetMedPy is designed for large, flexible, and efficient network medicine and drug repurposing analysis, supports multiple distance metrics, offers different null models, enables disease module extraction and drug repurposing predictions. It is built for scalability and performance. It is customizable for tailored biomedical research needs. And one of the important things to mention is that it's not finished yet. So it is currently under development. It is under constant development. So it is installable in pip. It is also manually installable. You can find instructions and examples at the GitHub webpage. You can email me if you have any doubt or interest in this software. And we are planning to include many different other features that allow for more complex and for even larger and scalable problems. So this is essentially the team. This is a collaboration between the Northeastern University and the Harvard Medical School. Dr. Julia Menichetti from the Harvard Medical School is pretty much the director of this project. Of course, Professor Joseph Loscalzo and my mentor, Professor Barabási from the Northeastern University. They are fundamental in leading the group. Professor Joseph Loscalzo is a medical doctor, so he's our guru to tell us when our hypotheses as network scientists, physicists, and mathematicians are wrong. So he ground our work in the medical territory. Professor Barabási has a strong experience in network medicine and in network science in general. And these are my co-workers, and all of them are postdocs. Gordana from the Harvard Medical School is a computer scientist. She helped in the packaging of NetMedPy and in benchmarking the project, the package. Michael Sivek from Northeastern University. He's a chemist by training. He was one of the testers of NetMedPy. He also contributed in testing and developing the package. And Rodrigo Dorantes, he's a Mexican fellow also. He is one of the designers. He and I are the designers of the software, and I am the main developer. So this is pretty much the team involved in NetMedPy. The groups are bigger than this. They are very large. Actually, I don't think that I will have enough space to thank all of the people involved and all of the people that integrates the 3 different labs. But this is the main NetMedPy team. So thank you very much. I am open to questions, and I will be very gladly if you have any doubts. And thank you for your attention.
Thank you very much. Thank you very much, Andrés, for the wonderful presentation about the NetMedPy. Yeah, the session is open to questions from the audience. So please, you can use the chat or the Q&A, or even open your micro. Please go ahead with questions. No questions yet? Well, I'm always able to do a question.
Go ahead, Hugo.
Of course. So, Andrés, sorry. I have indeed a question that occurred to me while I was seeing your presentation, and it is about the physical chemical. I'm also a chemist by so it is about the physical chemical properties of the drugs. And what is the role of these properties in the drug repurposing outcomes that you may have in the NetMedPy, even if they are not included at this stage, if this will be included at the further stages of the development.
Yes, thank you. That's a very good question. So one of the properties of drugs, for example, is that they don't target from the different associated targets that are associated to a drug. The binding properties of the drug is different for the different targets. So we have primary, secondary, tertiary targets. This is something that the typical network medicine pipelines doesn't consider, so they doesn't consider the dosage, for example. But this is one of the things that we want to include. The way that we are planning to include is to assign weights, for example, for spreading information, for spreading the perturbations produced by the targets, given this difference in affinities, in binding affinities. So let's say that we start with different binding affinities for the different nodes, for the different targets, then we can simulate propagation processes that will diffuse this information through other nodes, through other proteins in the network until they finally reach the disease modules. So including those kind of properties can help identifying not only what are the necessary steps, but also how likely it is that the primary targets are the ones that we want actually to target the disease, because we don't want secondary effects, right? So one of the things that we want is to be able to distinguish between secondary effects and the main effect that we want to produce in the network. So this is one of the things that we want to include in the software in the future.
Okay, great. And thank you. So questions have arrived. So the first one is, do you know if there has been any case of success trying to repurpose a drug for cancer with this tool?
That's a very interesting question. So it is the information that we have is limited in that sense. Cancer is one of those complex diseases that are very hard to study because there are so many systems involved. There are so many genes associated to cancer. There are so many factors contributing to the disease that the signals that we get are frequently not very meaningful, or then they don't make a lot of sense. But even when we have clear signals, the part of the validation involves many different processes. As I said, they involve clinical and experimental validations. What I know is that using network medicine, there are different companies like Cypher Medicine, for example, that is using this pipeline to evaluate possible drugs to treat cancer, in particular pancreatic cancer. And we know also that there is another company in China that is using this framework to repurpose a new experimental drug to treat cancer that is based on genetic modifications. So yeah, there are drugs, or there are examples of studies that use network medicine for drug repurposing. How successful they are and how successful they're going to be, that's a different story. We will have to wait and see what are the results of those studies.
Thank you, Andrés. The next question, can NetMedPy help validate drug candidates before in vitro tests, or do we need other tools like docking?
Yes, that is a very...
Docking simulations.
Yes, That is a very good question also. NetMedPy stops at the prediction level. In order to validate the result, and this is because to validate, there are many different ways to validate, and we wanted to provide the users the flexibility to use many different kind of networks, many different kind of studies, not only drug repurposing, but other kind of studies. So we stop at the prediction phase, and then there are, yes, we need to integrate other tools for validation, such as docking simulations, such as enrichment analysis, for example. But yes, NetMedPy will only bridge the part of the data integration with the predictions.
Thank you, Andrés, again. More questions for Andrés? So far, we don't have additional questions. So, and since we are already with 50 minutes in this second hour of the webinar, I think I will close the discussion. And so, if there are no more questions, thank you, Andrés Aldana, for his presentation again in the RePo webinar. And
Thank you.
I will share just again this slide to close the webinar, to take the opportunity to thank again to Carlos Loucera and Andrés Aldana, the two speakers from today's webinar with two Python packages for drug repurposing, that I'm sure that will be very useful for the scientific community and for the medical community, and also for the patients when the results are turned into drug repurposing cases. So thank you very much again for the participation in the RePo webinar series. This webinar series will make a summer break during the next weeks, and we will come again in September with more webinars dedicated to the computational tools for drug repurposing. So thank you very much again for your excellent talks. Thank you also for the participants. We really hope that they gain knowledge for these talks and from these talks, and we are very pleased to have you all on this session. Bye bye.
Thank you very much. Thank you very much, Hugo, and thank you again to the audience. Bom dia a todos. Buenos días a todos, and good day to everybody.
Thanks to everyone, and bye.
Bye bye.